temperature. All right, let's get started here, you guys. Listen, what we're about to do now is look at all the different things that can affect enzyme activity. So one of the things that we're doing is something called substrate concentration. So the substrate was what again in the reaction? Which one? The reactant. Okay, but what was it? H2O2. So the substrate is H2O2, and when the enzyme is added, H2O2 will break down into water and what? Oxygen. And how will we know this happened? Because the oxygen is a gas, and it will produce bubbles. All you have to do is look for the bubbles, okay? And it's pretty fast. The reaction is really quick. Okay, so we're gonna test the effect of the varying amounts of substrate. So we're gonna add 3, 6, 9, 12, 15, 18 drops of hydrogen peroxide, the substrate, and follow it up with the catalase. Now, how many times did I say so far that the substrate is hydrogen peroxide? About 5 times? Do you know when the next time you'll see it? On the exam. Absolutely. It's a very easy point, you guys, on the exam. Okay, the other thing that we're going to do is see the effect of temperature on enzyme activity. So we're going to have it set at 0 degrees cold, 20 degrees room temperature, 50, 70. So now we're looking at the temperature's effect on enzyme activity. Well, let me think and remember chapter 5. Oh, you mean I have to remember 5? Yes. In chapter 5, when we talked about proteins, we said that the secondary structure of proteins is held by what?
Hydrogen bonds.
Hydrogen bonds. Good. And then we also said that we can do what to break the hydrogen bonds? Add heat. Isn't that what we're doing? That's heat. Okay. We also said that if you heat a protein, you will eventually do what? Lordson. When you heat a protein, you will eventually do what?
Denature.
Denature it. Break it, denature it. Very good. Now I have some people who say, I kill it. Now, if you kill an enzyme, you go to jail. All right? There's no such word as killing or dying. The scientific term is called denaturing. Why am I saying this? Because again, it's on the exam. And if you say, I killed it, I'm going to be like, oh, no, there's no killing anything here. It's denaturing. Yes, sir.
So, to put it into a human term, if I were to murder a person...
Oh, my God. Uh-huh.
Hey.
I denatured you?
I denatured you.
No, the cops will not believe you. No. You still go to jail.
I didn't kill the guy, just denatured him.
No. Let's stick. Let's stay here, please. All right. I want you here for the exam. All right. So when we heat an enzyme, we break the hydrogen bonds that hold the secondary structures. And you remember some enzymes also, or some proteins, hydrogen bonds also hold their tertiary structures. Remember that? We are really unfolding the protein, and we call that denaturing. We are basically destroying the protein's structure. And what did we say? When something loses structure, it loses function. So do you think this enzyme, the enzyme under 70 degrees, catalase, heated up to 70 degrees, do you think it's going to work? No. How will you know that?
Because it's too hot.
No, how will you know that?
It loses the structure.
It will be no bubbles. See, I told you, when you look at the bubbles, it tells you the enzyme is working. So my question is, how are you going to know that the enzyme is not working? It's not going to bubble. That's the whole idea. Okay, so remember in chapter 5, we talked about we can denature proteins using temperature. But we also said changes in pH can also denature proteins. So we're going to test that one out too. What was the third thing that we said that can denature proteins? Heat, pH, salt. Well, we're not doing that one, salt today, okay? So we're doing temperature to denature proteins and pH. Sounds good?
Yep.
So it's going to look like this. One of these temperatures, the enzyme is going to work really good. How we're going to know that? Because it's going to bubble. The other ones, it will not work. How are we going to know that? it's not gonna bubble. Are we okay so far? This lab, you guys, is very straightforward. All right. And then we're gonna do this. This one we're actually gonna do first, real quick. We're gonna test to see if blood and or yeast has catalase in it. That's the whole idea here. So how we're gonna do it? We have a glass slide up here, and I'm gonna put it here for you so you can record it. We're gonna put a drop of blood and a drop of yeast, and then we're gonna follow it up with hydrogen peroxide. Hydrogen peroxide is the substrate. And how are we gonna know if blood has catalase? It's gonna bubble. Now, if you're sitting in the back and you're completely not listening to me or not even looking up here, you are missing a lot, all right? So I can send videos to certain people. So I want you to please stand up and watch and look over here, please. Okay. Here's a drop of blood on the slide.
Oh my God.
This is sheep's blood. It's not my blood. Thank God. I'll bleed for you, but not today. All right. Let's see. And here's a drop of yeast. Maybe 2. Okay. Now, all I have to do, look at how much yeast they give us, and that's all I use. It's amazing. Now, all I have to do is use, what was it? Followed up with the hydrogen peroxide. So I'm looking for whether blood has catalase, whether yeast has catalase, whether if both of them have it or if neither one has it. Look for the
Bubbles.
good. Take a look at it. Take a look at it. Let the people behind you look at it so they can have a visual of it. What is your conclusion?
Both have it.
They both have it. Good. I'm glad you said that. You're gonna go to page number. Let me find it. Hang in there. No, I'm confused now. Okay, page number 63. And you're gonna quickly write a statement. And what statement would that be? Both blood and yeast have catalase.
Under conclusion?
Just write it there. You don't need to worry about observation or conclusion. Just some place that they got a lot of space there. The conclusion again is both blood and yeast have catalase, confirmed by the bubbles that are created. You really want to use all that space, guy. Good. I like that. Okay, listen. Were you ever you were a child? Once at a time when you were a child, you may have ridden a bike or ran in the street and fell down on your knees and scraped your knees. You hurt yourself. And you ran into the house screaming, "Mommy, Mommy," and you're crying. And Mommy goes to get this brown bottle that has this magic solution in it that when she puts it on the wound, it bubbles. And then all of a sudden you forget you're hurt and you start laughing. Actually, that's what I used to do. Like you're like, that is so cool, right? And the pain sets back in and you start crying again. Well, that was hydrogen peroxide. And why did it bubble? Because you have blood like this blood, like the sheep has blood. Oops.
Whoa.
Hang in there. Videotape that one. All right. Like the sheep. You're okay? The sheep has blood. You have blood. I can still use it. Use it one last time. All right. The sheep has blood. You have blood. So sheep has catalase. You have catalase. So what was actually bubbling is your catalase breaking down the hydrogen peroxide into what and what?
Water
And oxygen. Very good. Yes, sir.
Why
Why do you use it?
Yeah.
Why do you use the hydrogen peroxide? For wounds? To kill bacteria. So what happens is this. Some bacteria have catalase and they can fight the hydrogen peroxide. See? I have catalase. Good luck trying to kill me. You see? But then there are other bacteria that don't have catalase. You touch them with hydrogen peroxide, they're dead. But here, we're smarter than bacteria there. You know what we do to make sure they're both dead? We use highly concentrated hydrogen peroxide. See, the hydrogen peroxide that you buy is at a concentration that will kill both of them. The ones that produce catalase and the ones that don't. So you're okay. The whole idea is to avoid infection. That's the whole idea.
Is it better than alcohol?
To some, yeah, hydrogen peroxide is it's the best thing. It's the best thing. Very powerful. Yeah. Yeah. It's the best thing when it comes to killing organisms. It's one of the best things. They actually use hydrogen peroxide to wipe down machines right before they wheel them into the operating room, so that you don't get infected while you're in the operating room. So it's very powerful. Yeah. Okay, let's go on to the next one. The next one is the substrate concentration, the different kinds of substrates, the different amounts, I mean, of substrates. So what's a substrate? Is the hydrogen peroxide, right? So the procedure is asking us to take 1 ml of catalase and add it into each of these tubes, including the broken tube. So let's see, make sure I don't hurt myself there.
How do people extract the catalase from the organ?
There's a procedure for that. Pretty simple procedure.
Okay.
Okay, so it's 1 ml of catalase in each one, and it tells you to follow it up with 3, 6, 9, 12, 15, 18. Now, you're going to turn to page number 64, and you're going to start writing these things down. On the first column, it says number of drops. You're going to write 3, 6, 9, 12, 15, 18, like that.
In one line?
No, down the column, There it is. 3, 6, 9, 12, 15, 18. It increases by 3. Okay, listen please. On the next column, you'll put the data. Which data? Well, as soon as I add the hydrogen peroxide, there is catalase in there. The reaction will bubble. And depending on how active the enzyme is, the height of the bubble will change. You see? Now let's predict. The more active,
The more bubble.
the more bubbles. So the higher the column. Sounds good? So let's try it out. What are we adding? Hydrogen peroxide. Good. Let's do this here. Now, I want you to do something. Promise me. As soon as you see the bubbles, I want you to say, wow. Okay? Because that's as exciting as it's going to get. All right? Nobody? Nobody? No wow? No wow? There you go. Thank you. Thank you. I thought you would get excited there. Okay, you're going to start writing the numbers down.
Wait, that was 2
The
drops.
first one, it was 3. The first one is 14. First one is 14. Millimeters, yeah? So the tube sacrificed itself for you guys. Good.
Okay, you want to remove that? That gives me anxiety.
No, you're fine. I need to show you something.
I'm scared you're gonna hurt yourself, Dr. Saad.
It's all right. I've done this before. 1, 2, 3, 4, 5, 6. Thank you. Thank you. Just keep it going.
Oh my God.
There you
that
go.
sounds so
All right, 20.
Getting bigger.
All right, now the next one is how many? 9. 2, 3, 4, 5, 6, 7, 8, 9. It's not O, it's what?
Wow.
There you go. Just, oh wow is even better. There you go.
Oh, wow.
Let's see. 30. All right, what's the next one? 12. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12.
Is this exponential growth?
No, it's not going to be like that actually. It's going to taper off, hopefully. Let's see. 38.
Oh, now let's go down.
No, the previous one was lower than that.
Yeah.
Make sure you're writing the correct numbers.
expected it to go like a 40 or so.
Well, hang on. 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. We are now at 41. So Lordson is happy. We got the 40.
Oh my, this is so exciting.
All right, 18. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18. Thank you for that. Thank you, thank you. Keep doing it, keep doing it.
Wait, you didn't measure.
I know, I will. Hang on.
My God.
It is gonna be 50. Yeah, 50. Believe me. Watch this. Watch this. Watch this. Here's your graph. There's your graph. You see it?
Oh,
When you eventually graph it by next week, well, you're gonna graph it hopefully right away when you get home, but you hand it in next week, it's gonna look just like that.
Wait, does it have to be a bar graph or a line graph?
No, it's gonna be all your graphs are line graphs. I'm glad you asked me that. Line graphs. It's gonna look exactly like that. Now, the interesting thing is this. Had I kept it going, it will eventually, listen to this one, it will eventually taper off. See, the more substrates I added, the more enzyme activity there was, right? But then it's gonna get to a point where if I add a ton of hydrogen peroxide, you will not benefit from it. Let's say, for example, we get to 60 millimeters. It may then, after that, stay at 60, 60, 60, 60. I don't care how much hydrogen peroxide you add after that. We call that saturation. Enzyme saturation. For you, you would say, I'm overwhelmed. Do you see that? Let's say you do 10 things every day. That's a lot of stuff to do every day. And I throw 15 things at you. You can only do 10. What's going to happen to the other 5? You're not going to do them. Are we catching the analogy here? You're only able to do so much on a daily basis. If I add more, you're going to feel overwhelmed. Well, that's the same thing. The enzyme gets overwhelmed, but we don't call it overwhelmed enzyme. We call it saturated enzyme. Beyond a certain point, it can no longer help you because it's doing its job at the maximum capabilities that it can. It can't help you anymore.
Wait.
Unfortunately, we don't have that yet in today's data. It's almost saturating, but had we done 3 more, it would have leveled off. All right? Yes, sir.
Considering that our protein can be saturated and be overwhelmed, what would happen if you add too much?
Too much what?
Too much hydrogen peroxide.
It doesn't matter because the enzyme can only work so much for you. But if your mitochondria is spilling out a lot of hydrogen peroxide, but the enzyme is not getting rid of it, then your cell is going to be poisoned from the hydrogen peroxide. Do you catch that? If the factory is producing a lot of waste, listen, if the factory is producing a lot of waste, pay attention with me please, that you cannot keep up with, then the waste is going to build up.
And you're going to get killed by...
Well, then you're going to suffer the consequence of that. But if every time the factory produces a barrel of waste, someone comes and picks it up.
No.
Someone that comes up, then you're okay, then you're fine. It doesn't accumulate.
I'm saying the hydrogen peroxide is concentrated when we bought from the store, right? So when we use it on our wound, it makes sure the bacteria is killed.
Yes, that's a different scenario. That one, we intentionally use hydrogen peroxide that is concentrated to overwhelm the bacterial catalase, to kill them.
What about...
Well, we're saying the same thing.
What about our own catalase?
If you overwhelm your catalase, your cells will be killed by the hydrogen peroxide inside of your cells. On the wound, temporarily, that's fine because your skin will recover from it and it will heal. It's part of the wound healing process. Okay?
Oh, okay.
But it's a price you pay for keeping your wound clean. You see what I mean? Knowing that your skin will eventually heal itself and you'll be fine.
So don't use peroxide.
No, use the hydrogen peroxide. No. All right. Okay. The next one is the temperature one. Now you may not have noticed me do this, but I'll tell you, I did this. I set this up while you guys were taking the exam, and it's these guys right here. And it tells you that you're gonna have one tube with a 1 ml of hydrogen peroxide and another tube with 1 drop of catalase. I have them right here. And I put them at all the different temperatures. There's water baths on the side of the room. I put them at all the different temperatures that are required for about 10, 15 minutes or so. Then I took them out and now they're here. All I have to do now is take the 1 ml of hydrogen peroxide, add it to the catalase, and look for the bubbles. Good? All right, here it is. This is 0 degrees. Nothing happened. So we're gonna go to page number 65. Here are the temperatures going down the column. 0, 20, 50, 70. Those are the temperatures going down the first column. The first result is it's 0. Good. What is your prediction for this one? 20 degrees. Will it bubble or not?
Yeah.
Yes, I think I think so. Oops. Wow. There you go. Thank you for that. All right, I'm going to measure this one here so you can get the data for that one. That is 55. Okay, what is your prediction for 50? Will it be more than this or less than this?
Yes.
It'll be less. You know why? Because it's on its way to denaturing. It's 50 degrees. It's on its way to denaturing. Let's see.
What? How are we gonna measure that?
I see it. It actually stayed around the same 55. That's unusual. It stayed at 55 too. All right, so it did not denature all the way. What's your prediction with this one? Flat, 0. I hope. All right, let's see. Thank God it did not embarrass me here. Okay, good. 0.
Wait, we forgot to say wow in the previous one.
It's okay. I'll forgive you. All right. Pretend as if the rest of these all popped, but the top of it is right here. And there's your graph right there. It should not have done this with this one. It should have been the highest here. So it should have produced a bell-shaped curve. But you graph what you get. All right. So it is kind of bell-shaped. It's kind of like flat on the top of the bell there. Let me move the mouse for me real quick. Do you see the mouse? Oh, here it is. Okay, hit the. I gotta do this password.
I don't know why that was so funny.
And I missed that.
Caps lock is on.
Thank you.
Best teacher. I know the password.
Good. So, did you get the picture of that? Okay, good. So you're gonna graph this. Now listen to me, please. Let's assume that this temperature had the highest activity. There's a name for that, and it is called optimal temperature. Optimal. Optimal. The optimal, the best.
Optimal.
Okay, here's the definition of optimal temperature. The temperature at which the enzymes work the best. This enzyme in particular. The temperature at which the enzyme worked the best. That is called the optimal temperature. So can someone tell me, what is the optimal temperature of all of your enzymes? 98.6 Fahrenheit, or what Celsius?
33.
So 37. Very good. Because that's what your body is set at. Your body is set at 37 degrees Celsius. All of your enzymes work the best at 37 degrees Celsius, and that's why you maintain your temperature at 37. Except for the men in the room. Listen to this one.
Oh, yeah, they
Men in the room, because of the sperm production, it requires a lower temperature, slightly lower. A sperm cannot be produced at 37 degrees Celsius. It's too hot. It's just too hot. The enzymes don't work. I should have asked you why. The enzymes don't work.
It's 95 degrees Fahrenheit, right?
Yeah, it's a little lower. So it doesn't work at 37 degrees Celsius. It works at a lower temperature because of the enzymes. They don't work at 37. That's why the testicles are technically outside of the body in the scrotum. They're not inside the body. There are men that are born with testicles that have not descended. They're going to be infertile. They can't have babies because it's too hot. Just a slight difference in temperature means a lot in this case.
can't fix that.
Huh?
They
I
can't
mean,
fix
unless it goes
No, if it has not descended, they can't fix that. No. That's just kind of, it's sort of like a birth defect, yeah. And some of them have one descended, some will have the one did not. Now, there are some men, they are infertile. The silliest reason is because they wear tight clothes. Listen to it. Some men wear tight jeans or whatever, and it's so tight that it kind of like positions them where the body is hot, and it gets too hot, and they can't produce the sperm. So what's the solution for that? That one has a solution. That solution is literally the doctor will tell the person to take a cold shower, meaning sit in a cold water bath for a while before they attempt to have babies. You see what I mean? And I'm not kidding you. All it is to reduce the temperature to start producing the sperm so that you can be successful there.
So the males that can't like produce sperm, they don't have sperm at all.
In some the men that have high heat temp, yeah, they won't have
No, I'm talking about
they'll
the
have.
ones with the testicles that
Oh yeah.
they don't produce sperm at all.
No, and even if they do, they're not gonna be functional, viable. Yeah. But some men have one testicle descended, the other did not. Yeah, does not.
That's when we dangle.
Yeah. All right.
Wait, so like, what if you start at 2, but you got surgery and got 1, so now.
It depends on how far low it is. If it's all the way inside here, the doctor is not gonna help you.
That's the question. It's gotta be
Yeah.
eat those people.
Yeah. Anyways, let's move on. That is not on the exam. That's just a side note there. Man, this is what it takes to stimulate you guys. Very good. Now I know where the button is. All right. Good. So now we know that at 70 degrees Celsius, what happened to the enzyme? It denatured. Very good. Good. Let's do the pH. The pH ones are over here. And write down the numbers: 2, 4, 7, 9, and 11. It's in the next table. 2, 4, 7, 9, and 11. And it asks me to do 1 ml of hydrogen peroxide in that pH. So this is what we're adding. The hydrogen peroxide in that pH. Okay, so 1 ml right here.
Acid right there.
Yeah, pH 2 would be acidic. So is pH 4.
So if you touch it, would it run off your hand?
Well, maybe. You're gonna feel the pain, and you're gonna run to the sink to wash it off. Unless you have very strong willpower there and watch your hand melt away. If you want to do that, don't do that, please.
Is it a nerve?
Why is it bubble on that
It
one?
makes you not feel like.
It's just an air bubble.
Yeah.
It's a trapped air. If you don't let that air out, you're not getting a 1 ml.
That's not healthy though.
You gotta get rid of that. All right, what do we follow it with? Catalase. Please pay attention, please. Please pay attention, please. Now, pH of 2, let's see what happens. Now, this is tricky because I have to get rid of that air bubble because I need to add just 1 drop. There we go. Didn't want to let go. All right. 0.
That's cool.
Nothing happened. It's supposed to be 0.
Like 1 millimeter for like a split second.
No.
There's one. Thank you for
Wow.
that. Thank you for keeping the excitement going. All right, 35. 35.
My goodness.
Okay, what's the next pH? 7. What's your prediction? Will it be higher than 35 or lower? Higher or lower?
Lower. Take my bet.
It'll be higher.
Wow.
You have catalase, you guys. Ooh, that did not do well. You know what happened? It fell on the side.
Hey, I was wrong.
I'm repeating
Where's
that
my
one.
money?
Can you get me a... Oh, I have it too, bro. Sorry. Thank you. I'm repeating that one. It fell on the side.
What? Reroll? That's cheap.
Reroll. No.
Was that one just water?
Let me try it again. This is the 7, right?
That's water, right?
No,
That's
not water,
no, it's
that's
hydrogen peroxide,
hydrogen
pH 7.
I won my bet and then they got reroll. Cheating, man.
Yeah, when it hits the side, it doesn't work well, of course. Let's go to 60. Thank you.
Oh.
Oh, catch it. There we go. This is pH of 9. And that one is... went back down to 49.
Oh no.
Now pH 11 is weird. pH 11 is supposed to be lower, but sometimes it acts really weird.
Why is that?
I'll tell you. It is lower. Thank God.
Sometimes the bubble is bigger.
41. pH 11 is not enough to denature this enzyme all the way. And I've been trying to get this set up at 12 or 13 actually works better for this one. But you don't need to know why, okay? I mean, it will blow your mind away, so we don't need that. All right, look at. Here's the graph. There it is. It goes up and comes back down. The best is at pH of? So if this was the optimal temperature, what would this one be? You got it. Optimal pH. The pH at which the enzyme works the best. Yes, sir.
So that means hydroxide or hydrogen ion concentration has something to do with that as well.
No. It's just a pH of 7. Don't worry about the hydrogen and hydroxide. Do that. The hydrogen and hydroxide are equal, so they're not hurting the enzyme at all. It's the hydrogen on this one is hurting the enzyme. The hydroxide in pH 11 is hurting the enzyme by denaturing it. Yeah? The hydrogen and hydroxide doing nothing to the enzyme at pH 7 because they're equal and they're not affecting the enzyme. Why? Because the enzyme is active. It likes pH 7. Now, for pH, the enzymes like different kinds of pHs or proteins. They like different kinds of pHs depending on where they are. For example, we have enzymes in our stomach that work just fine at pH of 2.0, even though catalase doesn't work at 2.0, because catalase doesn't work in the stomach. So, in our stomach, to digest our food, these enzymes work beautifully at pH 2.0, because that's where they live, in acidic environments. Right outside of our stomach, in our intestines, the pH is more basic. So those enzymes will work better near a pH 8.5. So now, for temperature, all our enzymes work at 37 degrees Celsius, except the exception that I told you, for men. But for pH, it all depends on where they live. If the enzyme works in the stomach, it'll be acidic. It will like acidic environments. If the enzymes work in the intestines, they will like it more basic. Okay? Your lecture, your PowerPoint tells you a little bit of that. Okay, so 1 week from today, for those people who are not here, you have to fulfill this lab exercise here. With the data, you will have to graph, and by the way, what I'm saying is for you guys too. Listen, you have to use the full size of the paper, of the graph. I have students who graph all their data in this little tiny square right here. I have to put the lab manual under the microscope to see it. Here's the thing, I don't have a microscope at home. I'll mark you 0, okay? You need to use the whole real estate here, please, all right? All of it. And the way to do it is spread out the numbers. So what was the highest number of drops? 18. So watch this. Let's look, 18 has to be here. So you space it out evenly so that 18 is all the way here. And that will force you to use the entire graph. The highest number for the data, what was that? What was the highest number for the data? 50? Is that it? Okay?
Yeah.
Then 50 has to be all the way up here.
Wait, what? 60?
50 or 60?
Which one are you looking at?
The concentration.
Yeah, the highest was 50.
Okay. So if your highest number is 50, then the highest, I would put the scale up to 55. You see that? A little bit more than the highest number. If the highest number is 18, I would put the scale up to 20. You see? And that will force you to use the entire graph. Here's the other thing. You have to label the axes. Now, do you remember? What goes on the x-axis, the dependent or the independent variable? Independent. Which of the two is the independent? Number of drops. That means the enzyme activity goes on the y-axis. Watch this. This is always going to be your y-axis. You cannot flip this. And this will always be your x-axis. I'm going to hold your graph like I'm reading a book. I'm not going to flip it. All right, here's the other thing. You have to use units. You have to add units. So if you say enzyme activity, I'm like, okay, how did you measure that? I don't know how you measured that. Did you look at the light? Did you look at the bubbles? You looked at the height of oxygen bubble in millimeter. That's what you did. So you here, it says here, height of bubble column in millimeter. That's what you write over here. Well, the same thing with this graph. By the way, at the end of the graph, you make a statement, a conclusion statement at the bottom. The same thing with this one, but this one is, what's on the x-axis for this one? Temperature. And then on the y-axis, and the y-axis, enzyme, enzyme activity, measured in height of bubbles in millimeters. Again, spread out the numbers so you use the entire graph. Make a statement. On this one, what's the x-axis for the last graph? What's the x-axis for the last graph? pH. The y-axis is enzyme activity, height of bubbles in millimeters. Now let me ask you this question. What will the maximum number be for the pH scale? 11 based on the data that we're using, but the maximum can ever be is 14. You know how many students bring me this graph here, and they have that the maximum number is like 50 or 25 or 30? There's no such thing. All right? If you want to label it from 0 to 14, I'm happy with that. But the highest was 11. So the bare minimum is 0 to 12. But no more than 14. You put 15 on your scale, I will mark the whole thing wrong because there's no such thing. Make a statement, please. You need to answer all of these questions here. You do not need to answer this one. Yeah, page 68. All right, thank you for that. Yes, sir.